day. This is the Professor, CBMD PhD, and today's Wednesday, July 5th, 2023. It is 3:03 and 20 seconds, AM Eastern Standard Time. And today's research, breast cells. So let's jump to it here. Largest ever atlas of normal breast cells brings unprecedented insights into mammary biology. Date June 28, 2023. Source, University of Texas. A new study has created the world's largest and most comprehensive map of normal breast tissue, providing an unprecedented understanding of mammary biology that may help identify therapeutic targets for diseases such as breast cancer. The Human Breast Cell Atlas used single-cell and spatial genomic methods to profile more than 714,000 cells from 126 women. The Breast Atlas highlights 12 major cell types and 58 biological cell states and identifies differences based on ethnicity, age, and the menopause status of healthy women. They were able to leverage many technologies to define, in a very granular way, all the different cell types and cell states in each of the main areas of the breast. They expect this tool will be highly useful for anyone studying breast cancer and other diseases such as mastitis, as well as breast development and lactation failure. The human body contains roughly 200 different cell types, 12 of which are found in normal breast tissue. Previous studies on breast tissue have focused primarily on epithelial cells. Given that these are known to give rise to cancer, but non-epithelial cell types have not been studied in depth using genomic approaches. Modern tools such as single-cell sequencing and spatial mapping allow the researchers to perform a highly detailed classification of 12 major cell type clusters, including 3 types of epithelial cells: lymphatic cells, vascular cells, T cells, B cells, myeloid cells, adipocytes, mast cells, fibroblasts, and perivascular cells. And for this study, researchers collected and examined 220 breast tissue samples from 132 women undergoing breast reduction or mastectomy surgery. Of these women, 46% were Caucasian, 41% were African American, 7% were Hispanic, and 6% were unknown ethnicity. And this wide-ranging dataset of normal breast cell types and their diverse states across females also takes into account individual factors, including ethnicity, age, BMI, obesity, menopause states, pregnancy, and a number of births, providing a wealth of information that serves as a powerful resource for the research community. As spatial mapping highlights 4 major cell regions, unexpected immune cell populations, spatial mapping techniques, which create a map of the cells within the tissue environment, allowed researchers to look at the RNA and protein composition of the samples to understand how and where the different cell types reside. And these techniques resolve the composition of known cell types and new cell states in the 4 main regions currently known in the breast, including lobular milk, producing areas, ductal areas that transport milk, connective tissue composed of fibroblasts, and adipose areas made up primarily of fatty tissue. The researchers were surprised to learn that 16.7% of all cells found in normal breast tissue were composed of immune cells, including the 3 major types: myeloid, natural killer, T cells, and B cells. Scientists previously thought that few immune cells would be found in normal tissue. Additionally, these immune cells were located primarily around ducts and lobules in 3 of the 4 major tissue regions. Understanding the nuances of these different immune cells can help in developing more effective immunotherapies for some subtypes of breast cancer and in defining their role in breast cancer progression. The researchers also found an unexpectedly high amount, 7.4%, of perivascular cells, including pericytes, which regulate blood flow from capillaries into tissues, and vascular smooth muscle cells, which regulate contractions of the arteries. Age, menopause, and ethnicity like roles in different cell types and cell states. The study revealed significant differences in breast tissue, composition, and cell states that were dependent upon ethnicity, age, and menopause status. As for example, African American women are disproportionately affected by aggressive breast cancer subtypes, such as triple negative breast cancer and inflammatory breast cancer, but little is known about the underlying cause of this disparity. The differences seen in baseline cell states and breast tissues from African American women and Caucasian women could, with further research, highlight potential markers for cancer risk prediction. Additionally, there were significant differences in the breast tissues for women over the age of 50 compared to younger women, as well as differences in cell states dependent on menopause status. The scientific investigators and researchers point out that more studies are needed to further understand the functional role of many of these cell states and to focus on other factors that may significantly advance the knowledge of human breast biology and disease. The Human Breast Cell Atlas project is ongoing and actively recruiting participants to build upon and improve the data sets, but the data is freely available now for researchers to access. And this is a professor of microbiology, bioinformatics, pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.